What's up, everybody? It's Miles here from bootcamp.com, and we're going to start our adventure into the study of pulmonology. First off, we're going to go through the basics, then we're going to work our way into physiology, and finish off the series by talking about pathology and pharmacology. So, without further ado, let's take a deep breath and do some respiratory. So today we're going to be doing lung anatomy, histology, and function. So first we're going to get into respiratory system function, then we're going to move into lung anatomy and histology, and finish off with embryology. So the respiratory system can be functionally and anatomically divided into the conducting zone and the respiratory zone. So functionally, the conducting zone is the first portion of the respiratory tract, and it conducts air down to the lower portion of the respiratory tract known as the respiratory zone. It pretty much only conducts air and does not participate in gas exchange, whereas the respiratory zone is the lower portion of the respiratory tract, and it does participate in gas exchange. Anatomically, the conducting zone goes all the way down until we reach a structure called the respiratory bronchioles. Once we are in the respiratory bronchioles, we are now in the respiratory zone, as the respiratory bronchioles and the more distal alveoli can participate in gas exchange. And the reason why the conducting zone cannot participate in gas exchange is because it has this very thick epithelial layer, and it's so thick that the gas can actually not pass through it, whereas the respiratory zone has a very thin wall to it. And so it actually does allow for gas to be exchanged from the respiratory system to the blood, and from the blood to the respiratory system. Now, the conducting zone doesn't just conduct air. It also has three other functions. So it has these blood vessels that run deep to the epithelium, and those blood vessels are going to contribute some heat to the air that passes through the airway. And so the conducting zone warms the air. There's also a thin layer of water and mucus on the epithelial lining, and that water is going to contribute some humidity to the air. And so the conducting zone also humidifies the air. And lastly, there's also these little hair-like projections coming out of the epithelium called cilia, and those trap foreign particles. And so the conducting zone also filters air. All right, now let's jump into some respiratory anatomy. So the respiratory system is divided into an upper respiratory tract and a lower respiratory tract. And so let's go over some of these structures. In the upper respiratory tract, we have the mouth, the nasopharynx, the pharynx, and the larynx just up to the vocal cords there. And in the lower respiratory tract, we have the trachea. We can see the trachea there. We can see it here. We have the trachea here, and it branches into the bronchi. And the bronchi are then going to subdivide into the bronchioles. And when the bronchioles start to have these little outpouchings on them of gas exchanging surface, we now call those the respiratory bronchioles. And then the respiratory bronchioles give way to the alveolar sacs. All right, let's move on to some respiratory histology. So we'll start off here by talking about the trachea and the bronchi. So the trachea and the bronchi have a special epithelium called respiratory epithelium. And the respiratory epithelium has a special description to it. It's said to be pseudostratified because if you take a look at the nuclei here, they're all in different planes. So it looks like they're kind of stacked up on top of each other. But in reality, each nuclei corresponds to one cell, and each cell is adhering to the basement membrane. So it's not actually stratified, it's pseudostratified. The cells are also ciliated, as we also talked about. There's these cilia coming up the top of the cell here. And there's also these goblet cells. We can see a goblet cell right here, and they are mucus-producing cells. The trachea and the bronchi also contain cartilage. And the cartilage are these supportive rings that support the airway and make it such that they are non-collapsible. So there could be an external pressure on the airway, but it will not collapse the airway because they contain this cartilage support. So they are non-collapsible. All right, moving on now to the terminal bronchioles. And so the terminal bronchioles are essentially made up of smooth muscle. And so because they do not have that cartilage support supporting them from external pressures, they are collapsible. So they could collapse due to external pressure, or it could be that there is smooth muscle intrinsically in the wall of these airways, which could contract and collapse the airway that way. The terminal bronchioles also have club cells, and those club cells will produce a surfactant-like substance that will coat the airway and help to keep them open. And they also contain some ciliated cells as well. All right, next let's move on to the respiratory bronchioles. And like we kind of mentioned before, the respiratory bronchioles have these little outpouchings of gas exchanging surface. And so they're a little bit more distal to the terminal bronchioles, and it's where the beginning of gas exchange occurs. But the majority of the gas exchange is going to happen in the alveoli. And so finishing off here with the alveoli, the name of the game for alveoli is gas exchange. This is where the vast majority of gas exchange occurs. And there are 3 high yield cells that you need to know in the alveoli. The first is the type 1 pneumocytes, and the type 1 pneumocytes are essentially the wall of the alveoli. So it's the wall of the alveoli here, and they're these very thinly spread cells. So in a histological image, we can kind of see these very long, thin cells that make up the walls. Those are the type 1 pneumocytes. And they're so thin that they allow gas to be exchanged in and out of the alveoli. Next, we're going to go over the type 2 pneumocytes. And the type 2 pneumocytes are very high yield. There are these large cells that live in the corners of the alveoli. So they look like these large cells here, and they live in the corner of the alveoli. And they have 2 important roles. The first is the production of surfactant. And we're going to be talking about surfactant in detail in the physiology video. But essentially, it helps to keep the alveoli open and prevents them from collapsing. The next is that type 2 pneumocytes are involved in regeneration. This is super high yield. The type 2 pneumocytes are involved in regeneration. So whenever there is injury to the lungs, the cell that's primarily affected is the type 1 pneumocytes, and that's because there's so many of them. They make up basically the entire alveoli. So the type 1 pneumocytes are going to be affected. So how do we replenish or regenerate these type 1 pneumocytes? Well, the type 2 pneumocytes can actually proliferate and give rise to new type 1 pneumocytes. So classic question, whenever there's lung injury, what cell is involved in repair or regeneration of lung tissue? And the answer is always going to be type 2 pneumocytes. Next, there is the close relation of the alveoli to capillaries. And that's because gas exchange needs to happen. So there can be gas exchange from the alveoli into the closely related capillaries and from the capillaries into the alveoli. Lastly, there are macrophages that live in the alveoli, and they're kind of located in the center of the alveoli. And they're responsible for phagocytosing any material that makes it all the way through the respiratory system, all the way down into the alveolar ducts and the alveoli. And so really the only thing that's going to make it down there are going to be very fine particles like dust, hence the name dust cells. So a macrophage in the lungs are called dust cells. And so there's a classic boards question that an individual inhales a medium to large particle and it gets lodged in the terminal bronchioles. What is responsible for clearing that particle? Well, the answer is going to be ciliated cells. It's not or mucociliary clearance. It's not going to be macrophages because macrophages are located deeper down in the respiratory system, in the alveolar ducts and in the alveoli. Now, if they have a question where the individual inhales dust and the dust is lodged into the alveolar ducts or the alveoli, as soon as they say anything alveolar, then the answer is going to be macrophages because that is the primary cell involved in clearing foreign particles in that area. All right, let's finish off with some lung embryology. So we talked about some different parts of the lungs, and now let's talk about how it develops. So the first stage is called the embryonic stage, and it occurs from fertilization to week 4. So recall from embryology that there is a structure called the foregut, and I have the foregut drawn here in orange. And the respiratory system is actually derived from the foregut. The lungs bud off of the foregut, and that is called a lung bud. And I have the lung bud coming off the foregut here in blue, and then that eventually is going to form a very primitive looking respiratory system like that. And it's at this stage that we can see an embryologic defect emerge called a tracheoesophageal fistula. And we're going to be talking about that defect in the next video. Next, we have the pseudoglandular phase, and that happens from about week 5 to week 16. And it's called pseudoglandular because at this point, the lungs actually kind of resemble a gland. You have the opening at the top and the glandular structures underneath. And at this stage, we get the development up to the terminal bronchioles. Next, we have the canalicular phase, which occurs from week 16 up to week 26. And it's called the canalicular phase because now we're developing all of these structures called alveolar ducts, and it kind of forms all these little canals, these small canals within the respiratory system. So we're going to develop the alveolar ducts, and recall that right before the alveolar ducts is the respiratory bronchioles. And so now we have some gas exchanging surface, and so respiration is possible at this stage. And we also have the formation of the type 2 pneumocytes at this stage. Next up, we have the saccular phase, and that happens from week 26 up until birth. And you could pretty well guess why this phase is called the saccular phase, and it's because we are forming all these little tiny sacs on the respiratory tree now. And these little respiratory sacs are the primitive alveoli. So the primitive alveoli form in the saccular phase. All right, lastly, we have the alveolar phase. And this actually occurs from birth up until age 10. So we can see in the alveolar phase that the lungs become a lot more mature, the whole respiratory system becomes a lot more mature, and the alveoli become a lot more developed and functional. And one of the keys to this stage is the fact that it doesn't end until about age 10. So in a board's question, if they ask you, when did the lungs stop developing? Is it going to be week 15 or week 24, etc.? The answer is actually going to be closer to age 10 because the alveolar phase, the final phase of development for the lungs, doesn't end until about age 10. So that's all I got for today. Hope you enjoyed the video and join us next time for embryologic defects.